Welcome back to Everything Explained in 5 Minutes, the podcast that takes today's biggest ideas and boils them down to the essentials fast. I'm your host, and today we're tackling one of the most powerful tools in modern biology, CRISPR gene editing. By the time you finish your coffee, you'll understand what CRISPR is, how it works, why it matters, and where it's heading in medicine. Let's dive in. 1. What is CRISPR? CRISPR, short for clustered, regularly interspaced, short palindromic repeats, was discovered as part of bacteria's immune system. Bacteria capture snippets of viral DNA and store them in their genome. When the same virus attacks again, they use those snippets to recognize and destroy it. In 2012, scientists realized they could harness the system, using a guide RNA to direct a molecular scissor, called Cas9, to any DNA sequence they choose. That means we can cut, remove, add, or replace genes with unprecedented precision. 2. How does it work? There are 3 main components. Guide RNA, gRNA, matches the DNA sequence you want to edit. Cas9 enzyme acts like molecular scissors, making a cut at that exact spot. Repair template, optional, a piece of DNA that the cell uses to fix the cut, and in doing so, incorporates new genetic information. Once the DNA is cut, the cell's own repair machinery springs into action. If you supply a repair template, the cell will copy from it, allowing you to insert or correct genes. If not, the cell simply rejoins the ends, knocking out the target gene. 3. Why it matters. CRISPR's precision and relative simplicity have unleashed a wave of possibilities. Disease models. Researchers can create cells or animals with specific genetic mutations to study diseases faster and cheaper than ever before. Agriculture. Crops can be engineered for drought resistance or improved nutrition without foreign DNA. Gene therapies. For the first time, editing a patient's own cells offers potential cures for inherited disorders. 4. CRISPR in the clinic. The big leap is moving CRISPR from the lab into human treatments. Blood disorders. Trials are underway using CRISPR to treat sickle cell disease and beta thalassemia. Doctors extract a patient's bone marrow cells, edit the sickle cell gene, then reinfuse the healthy cells. Early results show many patients off transfusions and free of pain crises. Blindness. Inherited retinal diseases like Leber congenital amaurosis have seen success. In Phase 1 trials, patients showed improved vision after a single injection of edited cells into the eye. Cancer. Researchers are editing immune cells, T cells, to better recognize and kill tumors. These CRT therapies have had remarkable remissions in blood cancers. 5. Challenges and ethics. CRISPR is powerful, but it's not magic. Off-target effects. Sometimes Cas9 cuts the wrong DNA. Scientists are refining enzymes and guide designs to minimize errors. Delivery. Getting CRISPR into the right cells while avoiding the immune system is a major hurdle. Viral vectors, nanoparticles, and chemical methods are all under study. Ethical questions. Germline editing. Changes that pass to future generations remains highly controversial. Global consensus currently restricts CRISPR to somatic, non-inheritable edits until we understand more. 6. The road ahead. Looking forward, we'll see. Base editors and prime editors. Newer tools that make even more precise edits, snipping single DNA letters without cutting both strands. In vivo editing. Direct injections of CRISPR components to fix genes inside the body, bypassing cell extraction steps. Expanded disease targets. From muscular dystrophy to metabolic disorders, dozens of trials are gearing up. In the next 5 years, CRISPR-based medicines could become routine, offered alongside surgery, drugs, and biologics. In 5 minutes, we've traced CRISPR's journey from bacterial defense to human therapy. It's a story of innovation, promise, and responsibility, rewriting the blueprint of life itself. Thanks for listening to Everything Explained in 5 minutes. Hit follow for more quick dives into the ideas that shape our future. See you next time.